I'm Makayla. I go to Northern Illinois University. This summer I worked in the law lab studying the role of CAT and BAP in immunity regulation. So to start off, CAT stands for catalase, which is found to be produced in plants under stress, and they produce reactive oxygen species. BAP is a bond 1 associated protein. It's in this giant protein family that contains BAP1, which we're going to talk about today. We're interested in BAP because it seems to be regulating plants under that stress stimuli, and it regulates cell death. So we're focusing on those two. Another study did this in grapevine, which is Vitis vinifera, where they overexpressed BAP1, and they saw that the enzyme seems to be reducing reactive oxygen species when they do this in Arabidopsis. So our question with all of this data became, does overexpression of catalase rescue the BAP defect when we do this in Arabidopsis thaliana? So the first thing we did was look at protein localization using green fluorescent protein, which we see here. In overexpression CAT in the wild type background, we see the green fluorescent protein localized around the membranes and the little dots, which we call puncta, which are cell organelles. Similarly, in the BAP mutant background, we see the same protein localizations showing us in both backgrounds proteins, the CAT is going to the same place. So the next thing we did was to look at the relative gene expression of catalase using a quantitative PCR. We can see that our overexpression of catalase in our BAP mutant backgrounds has a whole lot of catalase expression. We can also see this visually when we look at these plant phenotypes, where the wild type plants are quite large. Our overexpression catalase plants in the wild type background are also quite large. And then our BAP plants are small. Our overexpression catalase plants in the BAP background are also even smaller. So that was a really interesting phenotype to see. The next thing we looked at was plant immunity with Pseudomonas syringae, which is a plant bacterial pathogen. So we dumped these plants into the bacteria, let them grow, crushed up the leaves, and then counted the colonies that we saw grow on the plates, and we ended up with this graph. So when we look here, we can see that our BAP plant background plants have quite fewer growth of the pathogen when we compare it to the plants in the wild type background. We can also see this same similarity in the overexpression catalase plants as we do in the normal BAP mutant plants. This is showing us that maybe overexpressing catalase isn't actually rescuing the BAP1 immunity phenotype like we thought it would. And the last thing we looked at was reactive oxygen species, where we can see similarities in our wild type and our BAP, but we can see a whole lot of reactive oxygen species when we overexpress catalase 2, and we don't see it when we overexpress catalase 1 or 3. So in the future, we want to study catalase 1 and catalase 3 a little bit more, but our big takeaway from this is that overexpressing catalase doesn't rescue the BAP1 immunity defects like we thought it would, but we want to make some more transgenic lines of plants just to confirm what we're seeing.
Thank you very much.
Yeah,
Awesome. Thank you for sharing this work.